Hello friends, this is Dr. Anuprita Watharkar from Department of Biotechnology. Today we are going to learn the concept of transcription in prokaryotes. RNA synthesis from template strand of DNA is called as process of transcription. During replication, entire genome is copied, but in transcription, only the selected portion of genome is copied. The enzyme involved in transcription is RNA polymerase, which is also called as DNA-dependent RNA polymerase. Here you can see the diagram of transcription. You can see here the coding strand, template strand, which are important components for the transcription process. You can see the direction of polymerization for the template strand is 3' to 5', while the nascent RNA is synthesized in 5' to 3' direction. Let's try to understand the basic difference between the template strand and the coding strand. If you see the function, template strand, it acts as a template for RNA polymerase to build a complementary mRNA strand, while coding strand, it contains the same sequence as of the mRNA transcript. If you see the directionality, the template strand is read in the 3' to 5' direction, while the coding strand, it runs in the 5' to 3' direction. RNA sequence, which is nascent RNA sequence, it is the complementary to the template strand, while it is exactly identical to the coding strand. The other name for the template strand, it is called as anti-sense strand, while the coding strand is also known as sense strand. Let's try to understand the steps of transcription. Initiation, elongation and termination, these are three main steps of the transcription. In initiation, there are again three steps, that is the formation of closed complex, open complex, and third one, that is the formation of tertiary complex. After elongation, there is a termination process, and termination is also involved two types of process. Either it can be a row dependent or row independent. Here you can see the diagram of transcription process, especially the initiation process. Here you can see the template DNA strand, and you can see the formation of closed promoter complex formation. When RNA polymerase attached to the specific promoter site on the DNA strand, it forms closed promoter complex. After that, there is a DNA melding at specific portion, and there is a formation of a transcription bubble, which is also called as open promoter complex formation. There is the actual initiation of the RNA transcript formation, and there will be the elongation process. After that, either can be a row dependent or row independent process occur, and the transcription get terminated. Here you can see the diagram of that particular transcription bubble, that is an open complex, and you can see the RNA polymerase is attached at the specific sites. You can see here the coding strand, you can see here the particular template strand, and you can see the RNA DNA hybrid region. Let's try to understand the initiation process in detail. The transcription is initiated by RNA polymerase holoenzyme from a specific point, which is called as a promoter sequence. Bacterial RNA polymerase is the principal enzyme which is involved in the transcription. Single RNA polymerase is found in bacteria, which is called as a core polymerase enzyme, and it consists of nearly 5 subunits: alpha, alpha hash, beta, beta hash, and omega subunit. This enzyme is called as core enzyme. The core enzyme binds to a specific sequence on template DNA, which is called as a promoter region. The binding of core polymerase to the promoter, it facilitates and specified by the sigma factor. In E. coli, the sigma factor, it is called as sigma 70 factor. Let's try to understand the function of all the subunits of RNA polymerase. Alpha and alpha hash component, they are involved in a chain initiation as well as interaction with the regulatory proteins. While beta and beta hash, they act as a catalytic center and they actually involve in the chain elongation. Sigma factor, it helps RNA polymerase for the promoter recognition, while omega component, it helps for the assembly of core enzyme. Now let's try to understand how what is meant by closed complex formation. Binding of RNA polymerase holoenzyme to the promoter sequence, it forms closed complex formation. The core polymerase along with the sigma factor is called as holoenzyme, RNA polymerase holoenzyme. In case of E. coli, promoter consists of two conserved sequences, that is a 5' TTGACA 3' at minus 35 element and at 5' T8TA80 3' at minus 10 element. So at minus 10 and minus 35 location, there are two conserved sequences are present. These sequences are upstream to the site from which transcription begins. Binding of holoenzyme to two conserved sequence of promoter, it forms closed binary complex. In some bacteria, the altered promoter may exist which contains upstream elements, and some may contain extended minus 10 elements rather than minus 35 elements. You can see here the sigma 70 factor of E. coli. It has a 4 region and it has recognized the particular promoter region, and it get bound to the promoter region. Now next step in the initiation, that is the open complex formation. After formation of a closed complex, the RNA polymerase holoenzyme, it separates 10 to 14 bases extending from minus 11 to plus 3, which is called as a melding. So that open complex is formed. This changing from closed complex to open complex is actually called as isomerization. Again the same diagram you can observe, and you can see here how the initiation process proceeds and the actual transcription bubble get formed after DNA melding. Let's try to understand the next concept, that is the tertiary complex formation. RNA polymerase starts synthesizing nucleotide and it does not require the help of primase. If the enzyme synthesizes short RNA molecules of less than 10 base pairs, it does not further elongate and it is called as abortive initiation. When the RNA polymerase managed to synthesize RNA more than 10 base pair long, it ejects to the sigma 3.2 region and RNA further elongates and exit from RNA exit channel. This is the formation of tertiary complex. Here you can see the diagram how the tertiary complex get formed, and you can see here the RNA exit channel where your RNA transcript will get exited. Next, try to understand the concept of elongation. After synthesis of RNA more than 10 base pairs long, the sigma factor is ejected and the enzyme move along 5' to 3' direction continuously synthesizing the RNA. The synthesized RNA exit from the RNA exit channel. The synthesized RNA is proofreads by hydrolytic editing. For this purpose, the polymerase enzyme backtrack by one or more nucleotide and cleave the RNA, removing the error and synthesize the correct one. The gray factor enhances this proofreading process. Pyrophospholytic editing, another mechanism for the removing of altered nucleotides. Next process is termination. There are two mechanisms of termination: Rho independent and Rho dependent. Rho factor, it is an ATP dependent helicase protein in bacteria that act as a termination factor during transcription. You can see here in the diagram, like Rho protein, it actually binds to the rut region, which is a specific site on the RNA. After the binding with the rut region, Rho move towards the bubble, and exactly by using the ATP energy, it pull away the particular RNA transcript from the complex, and it releases it from the RNA polymerase enzyme. Now try to understand the Rho independent. In this mechanism, transcription is terminated due to specific sequence in the terminator DNA. The terminator DNA sequence, it contains invert repeats which cause complementary pairing as a transcript RNA forms hairpin structure. It is containing the GC rich region, and because of that there is a formation of hairpin structure. This invert repeats is followed by the larger number of T base pairs, more than 8, on coding an A, that is adenine, on template DNA. The uracil will appear in the RNA and the load of hairpin structure is not tolerated by AU base pair, so the RNA gets separated from DNA RNA heterodiplex. In diagram you can see how the hairpin loop structure has been formed, and it get released from the actual DNA complex. Next concept, that is a row dependent termination. In this mechanism, transcription is terminated by row protein. It is a ring-shaped single strand binding ATPase protein. The row protein binds the single-stranded RNA at the rut site as it exits from the polymerase enzyme complex and hydrolyzes the RNA from enzyme complex. The row protein does not bind to those RNA whose protein is being translated. Rather, it binds to RNA after translation. In bacteria, transcription and translation occurs simultaneously, so the row protein binds the RNA after translation has completed, but transcription is still on. These are the references. Thank you.